Okay, yeah. So, Reason, great to have you on. I think maybe we just wanted to start by asking you, you have this amazing blog, fightaging.org. You've been writing for how many years now on Fight
Yeah,
Aging?
an embarrassingly large number. I think I started that in 2003 or 2004.
Yeah, I'm sorry to admit I have not read every post, but I'll
Oh,
have to
no,
get an AI to parse it all for me and give me the summarized version.
well, somebody is actually working on that. Somebody said they'd throw it all into a large language model and see what the output was. So I'll see. I mean, I'm in no hurry to do that myself. And there seem to be a lot of people out there who love doing this. So if one of them wants to do that with the corpus, then go for it. But of course, the way these things work, you no guarantee of accuracy in the fiddly details, and it's kind of the fiddly details that matter.
Right on.
Agreed.
We'll have to
So,
interrogate Reason GPT in the future.
well, maybe that will even work. Who knows? Though I suspect it would be just as likely to say completely outrageously silly things about the way biochemistry works, since these models don't really have a good understanding of facts, as I understand it. But
Yeah,
nonetheless, it's an area of interest. The point of Fight Aging was always, do the thing that gets people to talk about this. At the time I started it, there really wasn't an ongoing place where people talked about things that weren't supplements. The idea was if you at least have a dial tone where there's something going on which people can look at and say, wait, people are talking about this stuff, then maybe that was useful. And of course, that's irrelevant now with all the other forums and subreddits and all the rest of it. But it's still the case that I find it personally useful. And there are people in the industry who tell me that it's useful, and there are people outside the industry who tell me it's useful. Never by emailing me, though, funnily, only when I turn up at conferences in person. Do I receive that sort of feedback. But so I keep doing it.
Yeah, I've kind of always interpreted it as your stream of consciousness.
It's exactly what it is. I'm just at the moment working through the excessive amount of review that goes into doing the end of year posts that I need to figure out a more efficient way of doing. And it's educational in a way just to see where sort of unbiased scanning of the corpus as it comes out takes you. And definitely there's an awful lot more neurodegenerative research running through the feeds than I would say anything else. I think that probably just reflects the bias of the NIA and where their budget goes, honestly, rather than an actual sort of reflection of research interests, because you'd think that cardiovascular would be a bigger area of concern.
We're going to dive into the sort of imbalances. I think that's one of the major topics we want to get into. But Nathan's question, what he wanted to get at was, you call the blog Fight Aging, and so what's the win condition for you? What do you consider
The win condition for this is, I think, it started out, let's not die, because obviously I was young and naive and thought that on the time scale of the rest of my life, we'll get to that point. At this point, I think the story that don't die. At this point, the win condition would be do some good before you inevitably have to exit this world to cryopreservation, where good means help meaningfully advance the state of the field. And Fight Aging is sort of a multi-tool in that respect. It helps some people. It certainly helped me raise funding for my company. And at the end of the day, if I can point to something and say, hey, I helped make this better before I have to get vitrified, then that sounds like about the best that can be done given the situation we're in. Wherein, in fantasy land, if you had complete mind control over the sources of funding, you could probably do a lot better than that. But on the grounds that it's a human action problem, really, you have to convince people. And that's a bootstrapping process. And while everything's an exponential curve at every point, and you look at the last few years and you think, wow, that went really fast compared to the 20 years that happened before the past few years. And then, at any point in the process, you will think that. So 2040 will be pretty crazy.
It's very hard to predict.
2023. And I imagine that it will just be pretty crazy from the point. If we get 3 billion into Altos now, you can imagine what it's going to be like in 15 to 20 years from now. If the high point of 2010 was somebody put $1 million somewhere, right? So it will be interesting.
I mean, that was when we started this project of making the roadmap. The question is, who is the roadmap for? Is it for somebody of my age? Is it for you, somebody of your age? Is it for somebody who was born today? The roadmap looks very different, actually, depending on what age you are. And
Well, I
so
think
I
like
guess,
kids born today, I mean, if you're 3 today, or you're just getting born today, then I think you have a decent shot at living forever, for some definition of living forever.
mm,
Certainly, if you look at the way things are going, a 3-year-old right now, if nothing changed, they'd live to 100 on average. It looks like we're in a time of radical change where we're about sort of 5 to 10 years into the 50-year business cycle for first generation ways to affect aging. And the second one will take off, 20 years into that process. So by the time that person is 80, there will be pretty impressive rejuvenation therapies floating around. And if you can have pretty impressive rejuvenation therapies at 80, then the sky's the limit. Those of us who are 50 today, well, we'll have good old ages, but unless things speed up very radically, that's the limit to it.
I think one more question on that mental model of your roadmap. Do you consider cryonics to be a possible win? Like, or is it really just like a backup that you consider undesirable? Like, how do you think about that?
Well, in my mindset is that major surgery is undesirable. So against that metric, actually dying and undergoing major surgery and getting shoved in a container for some amount of time, yes, definitely not great. I'd rather be awake for that whole period. But I don't see that happening. I don't think we're going to get this going in time. You have projects like senolytics that are, quote, simple, unquote, in the sense that it's a single set of targets, a single problem, and you can devote an industry to that, and they will come up with something pretty optimal within 20 to 30 years that will clear out all your senescent cells, or some combination of approaches that are pretty optimal to that end. Then you have stuff like lipofuscin and stem cell populations and other forms of metabolic waste that I think are much, much bigger problems, in the sense that every single one of those is going to require a separate development project. And there's hundreds of compounds in there that could be important. There's hundreds of stem cell populations, all of which behave somewhat differently. All of that's problematic. That suggests that we're not going to make sufficient progress on that front in time to stop those things from killing us.
Yeah, can you go into a little more about just the nature of the problem that we're facing and how, like, how do you think of aging? How do you define it in your head, and
Yeah,
how difficult is it? Um,
yeah, and that's characteristic of a poorly defined field. I mean, I come originally from astrophysics, and astrophysics is very like medicine, especially around aging. You go out there, you go measure something, you get a number that's between 1 and 5, and everybody's like, okay, cool, it's probably around 2. And if your number is somewhere between 1 and 5, then your methodology is probably not completely stupid. And then let's theorize around this. And some guy will say, I think it's 4. I mean, I worked on neutron star magnetospheres at the time I was leaving that, and I checked back in on that 5 years ago, and to the best of my knowledge, it's not advanced in the slightest. The controversies are still exactly where they were, and the two sides are not getting to the meeting point, because you can't build a neutron star in your backyard and measure it. So that, your data is what it is, and people's models are basically fighting with each other. Evolutionary biology is much the same. It's just a big fight over models, and it's not going to get resolved anytime soon, because there's so much leeway to argue about the loose data. So aging is in the same position at the moment, where you could argue pretty much anything based on the current data. So we have a bunch of data. You have so much data on everything that's changing with age, but you can't relate it to each other because that's complicated. You're looking at a complicated system. So the model I try to use to explain to lay people is, okay, let's say you have one of those massive German art statues made out of metal pipes. That's the size of a building and has 1,000 metal pipes in it. Okay, how is this thing going to fall over if you just leave it out to rust? So who knows, right? Because rust is really simple. It's a simple process. But if you inflict that simple process on a complicated structure, it gets difficult to figure out what's going to go on. Aging is analogous to that, in the sense that it's a bunch of comparatively simple processes, acting on a very complex system. You can measure all this stuff, and you can measure what's going on in the cause department, like accumulation of senescent cells, pretty accurately. You can measure what's going on in the failing department, like mortality risk, cardiovascular disease, all of these things, pretty accurately. But you've no idea how these two join together, because the two sides starting on the working forward, not very many people actually start from the cause and work forward department. That's not very popular. They start from the problem, work backwards. And those two sides are just nowhere near joining the dots together. Won't be for a very long time. And in fact, we probably won't fully understand how aging progresses at a detail level before we've managed to produce meaningful rejuvenation. You could just start with the causes and treat them and then see what happens, which is the ideal approach. I'm very much of the SENS school. I came into this around the time that the strategies for engineered negligible senescence were being put forward and first propagated seriously, and the Methuselah Foundation was founded. And that's the sane way of looking at aging to me. It's very clear that we are not going to be able to decipher aging sufficiently well to actually make use of that information in a reasonable way before we die. What we can do is take the much better understood causes of aging and try and fix them. If you fix it and nothing happens, oh well. If you fix it and something happens, great. And there's only so many causes of aging, and we're not discovering more at a very fast rate. I mean, the last one came up like 10 years ago or something, and before that it was 30 years. So we're reasonably sure that we know what root causes of aging look like. And if we just keep hacking away at them, that's the best choice to fix the problem in a reasonable amount of time, or extend people's healthy lives radically in a reasonable amount of time and with a reasonable amount of effort. Unfortunately, this is not a popular course right now for reasons that we can go into.
Well, first, I to some degree agree with that way of thinking about the problem, but I want to challenge you a little bit about it and see what your reaction is. Things like the SENS categories and the hallmarks of aging, I also think of them as just a categorization system of the gerontological literature. It's not as if we've done a purely empirical look at what are all the molecular differences between an old and a young person. There's a bit of a narrative fallacy potential there. And there is actually stuff that we haven't found just because it's not part of one of the normal disciplines. And we haven't gone about this in a purely empirical way. Do you think that there's a possibility that there's like significant parts to aging that we don't know just because of an artifact of the way we do research?
So, if you could produce a map of all the differences between old and young, there's nothing you can do with it. You can't apply that map in any useful way because it tells you nothing about the processes that produce those differences. The only way you can do this is also understand what all the processes are, and I'm not optimistic about that because we live in an age of biotechnology and we don't fully understand the cell. And it looks like it's going to be decades before we can even come close to being able to say we fully. Question of, are there things we don't know? I think we should expect there to be things we don't know. For example, nuclear pore proteins. They don't appear to change very rapidly. They get damaged. And do we know how influential that is on aging? No idea. Because there are other problems that make it irrelevant in comparison, but that may or may not be the case. We have no idea. So we have all of these processes to look at, but again, because we don't have the full map, we don't know how important each process is. Senescent cells were always, from the start of SENS and ever since, a important point that people kept trying to convince the rest of the research community that these are actually important in aging, and slowly that fight was won. But everybody involved in this fight to convince the rest of the research community that senescent cells are important in aging has been surprised by the degree to which senescent cell clearance produces results in mice. Nobody was expecting senescent cell clearance to be as good as it is. And yet we're still at the point where we've no idea what it's going to do in humans, zero. We've no idea whether mice are extra sensitized to senescent cell burden in the same way that they're extra sensitized to growth hormone metabolism and calorie restriction with comparison to us in terms of effects on lifespan. So we'll see. And the only way to find out what the size of an effect is to go try it. And I think I've rambled off the topic here a bit, but it is certainly the case that there will be unknown unknowns. but that doesn't stop us from jumping in on the known things. And it seems to be that if you take the path of senolytics, right? If you take the path, a bunch of people said, wait, these senescent cells look really important in aging. Why are you not doing stuff? And then 10 years pass and somebody does something and it seems influential, and other people say, wait, we can do something. And then some companies get started 5 years after that. And then 5 or 6 years after that, there's some human trials that are sort of desultorily plowing along. But all the way along this, the mouse studies look amazing, absolutely amazing. The most impressive mouse studies of aging are all senolytics. Everything else is candy compared to everything else is just boring and trivial and not great compared to what senolytics can do in mice in terms of reversing diseases of aging very rapidly. I mean, even the standard of mTORC1 inhibition that reliably, robustly slows aging in mice. It doesn't reverse age-related disease in the way that clearing senescent cells do. So, 20 years in maybe to this whole exercise, we're still looking at another 10 to 20 years to get anything deployed into large chunks of the population. But this is the fastest, best path to go, sadly. How else are you going to do this? Are you going to sit around waiting for greater knowledge? Or do you pursue the things that seem like they continue to work and keep pushing on those studies?
Well, it depends on how many resources you have, right? You can sort of saturate a particular path potentially where you're into the diminishing returns, where spending more money trying to advance senolytics wouldn't be the best use of resources. Like you would want to spread it somewhere else. But we're in a situation where we have scarce resources right now.
Well, yes and no. I mean, if you're in a gold rush, you have infinite resources. Unfortunately, they just go into parallel. If you're in academia, then you're always going to be limited until such time as it's too late and it's pointless, at which point the powers that be will say, we should be doing more research on this thing that's now proven in the clinic. So senolytic research in academia will really take off after senolytic drugs become widely used. But the current situation is not ideal in many ways. You have 20 senolytic companies doing, of which maybe 5 are usefully relevant together, because different senolytics will do different things in different tissues, and we probably do need that exploration. But this is the nature of it. We're not good at collaborating as a species. We run in and do the things that are attractive, and it's not always necessarily sane as to which things are attractive and which things are not. I mean, for example, why has reprogramming become the poster child for reversing aging? Why not senolytics? Senolytics have far, far more impressive data. How is it that the reprogramming side of the house got the enormous backing of very high net worth organizations on the basis of far less impressive data? So it's kind of funny the way things pan out. But so long as there's money coming into the field for things that are not stupid, at the end of the day, it will move forward. And perhaps that's the best we can hope for as a species, that we are actually putting money into it, and there are actually people who think this is a good idea. And anything more than that is just pipe dreaming,
Yeah, well, I think that actually highlights one of the reasons why we're doing this podcast and building the roadmap, is because it does seem like we go through these hype cycles, and there's a lot of just historical of accident of who talked to which rich person when and what was their particular pet theory at the time. And to try to, for people that are new to this field, to try to get an overall sense of it, how we should sort of reasonably distribute resources. It's actually very hard to do that. Like to read, for instance, your entire backlog of your blog would take a lot of time. And so that's what we're trying to do here, is to try to help produce something that you could binge in a weekend and get like a sense of the field potentially.
Well, that would be good. I've so far shirked any responsibility I might have had to producing such a thing. My current thinking is that it takes about a year or two for someone smart to come in and get a sense of what the community is, where it's going, what it thinks. And in part, that's just because it's a complicated topic. in part because those summaries don't exist, in part because the natural unit of theories about aging is one aging researcher, and everybody thinks something different about the data. But it is, I agree, a tough problem. This is what I tell entrepreneurs who are coming in right now. Budget a year or 2 years, and if you're really enthusiastic about doing something faster than that, temper your enthusiasm, because you'll just run off and do something silly. And judging from the people I've seen come in and work with things and then go off and start a venture of some sort, it has been pretty universally 1 to 2 years in order to get a proper understanding and meet people and go to the conferences and get a grasp of the research and where it stands, all the rest of it.
Yeah, Nathan, you've actually been doing this longer than me in a sense, but trying to onboard people to this industry, I mean, that's what we do at the LBF, right? And trying to speed that process up and make it more efficient. Did you have any thoughts on that, Nathan and Sheila?
Yeah, I mean, it's one of those things where you have to plant the seeds early. I think one of the things we've seen through LBF and when I was previously at ODLB, right? It just takes time for people to really get a sense of the idea maze of where they can really have the biggest impact. But one of the things, Mark, as you mentioned, is we're trying to create this roadmap so that we can hopefully give people a broad overview first, so they can streamline this process. Because otherwise, it's kind of the same process every single time. A newcomer comes into the field and says, they ask the same question: what should I do? What should I work on? And then they talk to the same people, and have and you can only really go so far in a 30-minute Zoom call or something like that. To get really in the details, you have to
There's also the matchmaking problem in that there aren't infinite researchers and infinite research programs. There are only a set number of those that are at a point which makes where they're exposed and to the public, to a sufficiently large section of the public to be interesting, and B, licensable, and C, sensible in the sense of proceeding. And a part of that coming in and learning is actually trying to find who your scientific collaborator is going to be, or what project you're going to work on. And you can come in and have an idea as to what you ought to do. You could come in and, for example, say, I want to do thymus regeneration based on, and that's a sensible goal. It's clearly good, clearly useful. And then you can look around and find that everything sane already has somebody way further ahead of you working on it, which is probably the case for thymus regeneration, just looking at what the options are. So, I mean, you could upregulate FOXM1. You can try that with small molecules. You could try gene therapies. There's people out there who've done that. There's a gene therapy that may be licensable right now, but we tried it. It didn't do so well back a few years ago. There's the cell side of the house where you can deliver cells home to the thymus, and a very well-connected group is currently doing that. So I wouldn't try to go compete with them. And aside from that, the ground is there's companies basically already covering all the slots. So there's a certain amount of give and take. You can't come in with an idea and expect necessarily to be able to find a scientific partner that's in the right place for you to do something with a sensible approach. But equally, you can't be unguided, because there's just everything. It's a huge field. You need to have some idea as to where you want to focus. So it's a challenging trade-off.
Yeah, one thing I wanted to dig a little bit deeper on, going back to sort of your bigger overview mental model of how we could potentially get to indefinite lifespan extension. So I think the dominant sort of view that maybe that you share, in the SENS model or even hallmarks of aging model, is to develop interventions, drugs, whatever, to target these individual hallmarks or SENS categories, and then go for a intermediate indication, right? A disease indication. And then I think a lot of people also sort of think, okay, after doing the first disease indication, do these multi-morbidity trials, right? Something like a TAME trial to show delay in comorbidities of aging. And then I think the model is just kind of okay, then people just start taking these drugs, and then we'll just assume that the lifespan extension will just show up in the demographic data. Do you agree that this is the kind of like the going model?
Well, fortunately, I know enough about the model you're proposing to understand what happened when you froze in the middle there. But so and I will say at the outset that I'm stupendously disinterested in the mechanisms by which a drug gets from approval to widespread use, because I think that's a solved problem, and a lot of people work on that. It's a major focus of if you get a drug that works, then it's going to go places. I think the more interesting question is how do you get the drugs out there in the first place at all? Because as you point out, it's not actually the default assumption that one should go find a part of SENS, some form of damage, and repair it. There are companies that are doing this, but it's by no means a majority model in the community. This is still an industry of small molecule development, and it is still very much an industry in which the goal is to produce boring marginal therapies that do a tiny amount of something and get passed through on the basis of doing that tiny amount of good, and then make some pharma company money over time for 20 years or 30 years. So, I mean, if you look at all of the most advanced companies in the longevity industry, they are doing small molecules to adjust metabolism in the hopes that aging is going to be modestly slowed. And, at some point that has to stop, and people have to move on to something else. Because the thing to remember is that 100 years from now, nobody's going to remember that we were all taking a drug to slow our aging by 2 years. And two, as Brian Kennedy has adequately demonstrated, if you combine any two drugs that slow aging modestly, the chances are very good that you'll get a result that accelerates aging modestly. These drugs interact in ways that are non-obvious, and it's just not a good path. And this has always been the SENS-based objection to business as usual in the small molecule space. Small molecules have their place, and there's certainly ways they can be used to remove aspects of age-related damage, but adjusting metabolism is just not the right path to go. And yet it is the majority path that's being taken now, because medical research and development is 100% money oriented. And that's just the way of the world right now. Anybody who does something in the field that is not money oriented is one of the few unreasonable zealots. And maybe the unreasonable zealot succeeds, maybe they don't. But at the end of the day, the whole structure is determined by this goal of let's produce small molecules that can be taken permanently over time in patients. And whether they do good or not is actually completely irrelevant.
So I actually, this leads into, I was trying to find, at the Foresight Biotech workshop last year, you gave a really good talk on the problems faced in gene therapy. But I think one of my favorite quotes from that talk was just the way you were describing the limitations of small molecules relative to genetic medicine approach. And I guess, unfortunately, they didn't get a full recording of that talk. They only got like the second half. And so so I'm going to ask you now to attempt to recapitulate or see if you can remember, or at least give your current understanding of why are small molecules, you've already said a little bit, but why gene therapy is a better approach than small molecules, but then what are also the challenges with gene therapy?
Gene therapy is better because a small molecule only ever does a fraction of what a gene therapy can do. Small molecules usually can't affect they're not the protein machinery themselves. I mean, you can make a small molecule out of a part of the protein machinery, and there's some things you can do like receptor agonism, where you basically get the same result whether you deliver the full protein or whether you deliver a drug that does it. But generally speaking, a small molecule drug has a tiny fraction of the effect size that the genetic studies that preceded it do. Researchers prove things out using genetic manipulation, and then they try to find a small molecule that has 10% of the effect maybe, and then they try to make a drug out of the small molecule. So the future is you just make a drug out of the genetic manipulation, which will be one, far more effective, and two, it will not have any off-target effects that don't emerge from just altering the thing you originally wanted to alter, whereas all small molecules tend to have off-target effects, and issues with how plastic the therapy is. With a gene therapy, you can change it. You can change direction and change what you're doing quite readily and radically and easily. Small molecules are hard to re-engineer. That's difficult. Once you have one, you're kind of stuck in that direction and with its drawbacks. So that, in a nutshell, is why I use gene therapies. The downside is that small molecules have the distribution problem solved. Small molecules just go everywhere. You get them everywhere in the body, but gene therapies definitely don't. There are very few proven delivery options for gene therapies. You can deliver them to the liver, you can deliver them to the skin, you can, with some challenges, get them into the brain, and everything else is a major undertaking and not solved. So delivery is the challenge for gene therapies. And if you're a gene therapy company, basically you're a gene therapy delivery company. The gene therapy is the easy, trivial part. The hard part is how do you get those nucleic acids into the cells you want them into without getting them into the cells you don't want them into. And that continues to be difficult. And at the moment, everybody's stuck in a Goldilocks sort of scenario where lipid nanoparticle mRNA looks okay, and it's also kind of like small molecules in that you have to redose it a lot, and people are kind of interested in that because it's basically just taking the small molecule paradigm and applying it to genetic therapies. Or you're using AAV that lasts forever for the few rare conditions where people are okay with using AAV, or you're treating the liver, and nothing else is possible without an awful lot of work. That won't be true forever, and probably won't be true for 10 years, but right now it is a lot of work to develop a gene therapy. And again, from the financial point of view, nobody cares how well your therapy works. They just care that you can sell it and make money. So you wind up with these ridiculous situations wherein it is actually very hard to produce a better therapy for a condition that already has a bad treatment, especially if that bad treatment is a small molecule that is now generic. Because the position of the powers that be that have the money is, well, this is a solved problem. There's a treatment for this disease. We don't want to compete with a cheap treatment. It doesn't matter if your treatment's better. You will still struggle with it. Yeah, in that case, statins and atherosclerosis. Your treatment has to be absolutely amazing in order to even stand a chance to make headway in a market where there's already a treatment. So there's a lot of perverse incentives in this industry, and I don't know what the solution to that sort of thing is, except have some rich people fund development up to the point where you can offer it for medical tourism, and then get stuff out there for medical tourism, at which point other parties will start to play the game of introducing these technologies into regulated markets, which is kind of what happened for first generation stem cell therapies. I mean, the FDA and the EMA are organizations that typically only move when they're made to look like fools. So if really life-changing therapies become available outside the US, then that creates a track for them to be more rapidly and reasonably approved in the US. The FDA finds it very easy to say no, and investors find it very easy to believe that the FDA will say no to novel therapies that aren't already in existence somewhere else being demonstrated to be a fact. If you want to put something new through the FDA, it's just good luck. It's a real challenge. It's very expensive, and outside factors are almost more important than the efficacy of your therapy as demonstrated in something other than humans, which is why it's useful to go do first in human studies if you possibly can outside the US, just to demonstrate that what you have is more real, as it were, and make them feel like there is the possibility that this could be available outside the US if they're not going to be amenable to to showing it through the gauntlet.
Yeah, I mean, the FDA as an obstacle, I think, like, there's some pretty clearly true aspects to that, but there's also a bit of it's like humans get attached to certain stories. And so, like, the story of like small molecule development is the story of like sort of 20th century medicine because it actually solved a lot of problems in 20th century medicine when gene therapy was like way beyond the horizon. But then we're just attached to that way of doing things. And the FDA was designed to um, prevent harm, right, by people selling things that didn't actually work or that actually like or were actually like harmful. And so that's the story that that institution is built around. And it's like sometimes it's just changing the story. I think there can be a lot of resistance because people just become emotionally attached, or they're not even, or they're just subconsciously just going along with the story. And it's like, so it's like, yeah, doing something in a different jurisdiction, like forcing people to realize that, oh, actually, like we could be doing this differently. Um, there's probably a lot of ways that we could be doing that in the longevity industry because we need to change the way people think about healthcare, the way they think about research too. Um,
Indeed. I mean,
I,
at the moment, there's the strong danger that the lesson we're going to be giving to the rest of the world here is that the longevity industry is rapamycin and metformin and whatever BioAge is producing that is basically the same as that.
on whatever diet Brian Johnson is on, and
Yes.
things like,
You know, the story is get a few extra years, i.e., this is just the legitimate version of what Revlon has been saying for the last 20 years. And that would be a shame because we can do a lot better than that. And the drugs that are really amazing are like dasatinib and quercetin as a senolytic, are really not getting the attention they should get.
maybe we could segue a little bit into your own work right now, then, actually, speaking of things that could have a big effect.
Yeah, so we founded Repair Biotechnologies, Bill and I, to do something, and the decision to do something predated the decision to do something specific. So he talked me into starting a venture, and then we went around and looked at what we were going to do, and we settled on thymus regeneration. And then we took a year or two to demonstrate that actually no, the way we picked for FOXM1 upregulation, there was actually a good reason as to why the research community had tried that and then moved away towards other approaches or hadn't commercialized based on that. It's actually rather hard, and that comes back to the delivery problem of gene therapy. It's very hard to deliver gene therapy to small organs in any way that's going to be palatable to the community at large. I mean, you can directly inject the thymus, but nobody's going to be up for that. It's right over the heart, under your rib cage. It's not a good thing. So we along the way we found a really great technology that Aubrey introduced us to. Aubrey de Grey introduced us to a researcher who had found a way to selectively break down cholesterol in cells without actually harming the cells themselves. And this is actually really amazing because as you get older, cholesterol does tend to build up in various locales around the body. And if you're obese, then basically you have 10x that problem going on everywhere in your body. And cholesterol is a very weird molecule in the context of mammals in general because it's something that's needed everywhere. But because it's expensive to manufacture, energetically speaking, it's only made in a few places. So it's made in the liver, or you get some of it from the diet. And from there, there's this really Rube Goldberg, complicated, ridiculous system whereby cholesterol is altered and transported around the body and ingested by cells and put back into the bloodstream by other cells. And it's very silly. And like all complex systems, it's very prone to malfunction with age. See, as soon as this business of transport and how much do I have in my body starts to break down, then you get these localized deposits of cholesterol. And cells have some ability to say, no, I'm not going to take up that cholesterol, or I'm going to turn it into a safe form and keep it as lipid droplets. But it's not they don't have much of a buffer. As soon as you get even minimal excess of cholesterol in tissues, that buffer for dealing with it starts to get overwhelmed, and cells get free cholesterol inside them. And free cholesterol, the unmodified form, is basically explicitly toxic. If it's inside a cell and just hanging around, it is toxic. It's making the cell unhappy, and it will kill the cell. So this is what causes atherosclerosis. It's what causes major problems in the liver, in obese people, and honestly, it just causes problems in the brain and a whole bunch of other places. There's some rare conditions in which this is a problem. Obesity, almost everything to do with obesity has a sizable too much cholesterol aspect to it. And, arguably one could say, well, actually, cholesterol deposits maybe should be a hallmark of aging, though that might be stretching it a little bit. But they are characteristic of aging, because atherosclerosis is characteristic of aging, and that is localized cholesterol deposits, roughly. So we armed with this technology that we can use the comparatively limited methods of modern day gene therapy to get into cells in places that we care about, like the liver. We can then aggressively, rapidly turn back liver dysfunction and also atherosclerosis. We clear the lipids out of plaques, we reduce plaque size. And reducing plaque size is the thing that you can't do today with any of the present day technologies. That the challenge here is that the presence of excess cholesterol in a tissue is the problem. And all therapies aimed at atherosclerosis are aimed at reducing blood cholesterol. But blood cholesterol is only sort of lightly connected to how much you have in your tissue. It's not a very strong driver of that. Over time, yeah, kind of influential, but in the moment, it's not a very strong driver of that. The stash of cholesterol in a particular tissue that shouldn't be there, you can lower blood cholesterol to next to nothing, and that will do exactly nothing to that stash of cholesterol. It will sit there. And particularly in atherosclerotic lesions, because they're little toxic dumps of cholesterol on dead cells, the mechanism by which they grow stops being blood cholesterol and starts being attracting macrophages and killing them. So these things become macrophage graveyards, and there's liver dysfunction driving all of this in ways that are not connected to blood cholesterol. So all of this can be fixed, just given a gene therapy that shoves a fusion protein into cells, and that fusion protein just basically eliminates free cholesterol. And if you can just get rid of that free cholesterol, then a lot of things start to reverse. Atherosclerosis being one of them, metabolic-associated steatohepatitis being another one, a liver condition. And those two markets are vast and enormous. And, if going back to the do some good in the world, if I and the researchers that I'm working with can convince the research community more broadly and pharma that actually they should stop focusing on LDL cholesterol in the bloodstream and start focusing on local deposits of cholesterol, and hey, look, here's a way you can do this, that's good. Primarily because 25 to 40% of humanity dies from atherosclerosis, and
a lot of people already have a lot of the disease accumulation, and you can't, if your therapy only slows it, it doesn't help them.
And there's no blood cholesterol level that you can reasonably reduce to. I mean, there's a guy out there who's using PCSK9 inhibitors and a statin and some other weird supplement to reduce his LDL cholesterol down to 20. Where a normal very fit human is 70. He will still die from atherosclerosis if cancer doesn't get him. It's just going to be later. So that there's no lowering that you can reasonably do that's going to stop you getting atherosclerosis. You're still going to get it.
And this was the switch in thinking from like slowing to, or even prevention, to repair and rejuvenation.
Yeah,
that's
reversal
one of the
is
things
the
that
only,
not many people work on.
Yeah, reversal is the only game in town if you want to have large effect sizes. If you want to stop, because, so 10 to 20 million people are going to die from atherosclerosis. In the worldwide population of 10 to 20 million people are going to die from atherosclerosis in, let's say, the next year. And that's the addressable market if you really just want to go after this from a reversal perspective initially. But the view is that 30 to 40 years down the line, everybody's taking this drug in their 40s to stop from ever developing atherosclerosis in the first place. You take it at 40, take it at 45, take it at 50, that sort of thing. So the fatty lesions that are in people in half of the population in mid 40s just never form. So nobody ever dies from atherosclerosis ever again, which seems to me a useful goal to achieve. And certainly the research and development community needs a kick in the ass to stop them thinking that the only thing they can do is reduce mortality by 15 to 20%, which is what statins do, arguably. I mean, maybe not even that.
So I think your same mode of thinking can be applied to like a lot of different types of damage that accumulate. And I want to get into talking about other people's work and other people's strategies and your thoughts on them. But maybe one last thing on your own personal project is what is the bottleneck to you getting this available for humans? Like, or what are the multiple bottlenecks?
It's always money. That's the story. We proved this worked in the sense that, yeah, this looks pretty amazing. Now we just have to turn the crank. We got that stage like 2-3 years ago, and we've been turning the crank since then. Obviously, gene therapy companies, so that means delivery, trying various different delivery systems and migrating our way through the limitations of those and finding our way through to the final product that's lipid nanoparticle mRNA, rather than any of the other options on the table. But really, the thing that stops us right now, since we're at the stage of putting in pre-INDs pretty much, is money. Because if you're going to go take a lipid nanoparticle mRNA therapy to the clinic, let's say you just say do a phase 1b trial in 20 people with homozygous familial hypercholesterolemia, which is the genetic form of accelerated atherosclerosis, where they lack a functional LDLR gene, so the liver can't take up cholesterol, and their blood is milky, and so those guys. It's an orphan condition. It's the easiest way to get through the FDA, and you don't have to do huge trials. And in principle, your therapy will be able to be used off-label for sporadic atherosclerosis in other people, or at least ours would. So if you're going to do that, you have to license a lipid nanoparticle. You have to license some various other mRNA things. You have to do GMP manufacturing. You have to conduct your GLP studies in non-human primates and all the rest of it. And you'll easily spend $20 million to get to the end of get from I'm in late preclinical, this works, look at these mouse studies, to I have done my phase 1b and it works in people, and that will take you about 2 years. And of that money, pretty much 15 million of it is licensing and GMP manufacture. It's just a straight pass through to the various entities that are giving you technology, IP, and some support, and the entities that are manufacturing the drug to the standards required by the FDA. And I should say that those standards are vaguely sane in the state for something like a monoclonal antibody, where if you get it wrong, you kill people. But for lipid nanoparticle mRNA, I mean, come on, it's far and way overkill. Lipid nanoparticle mRNA type therapy is very safe. It's hard to get these wrong in ways that will kill people. If you're the FDA and you care deeply about 1 in 10,000 people having a reaction, that's one thing. But from the point of view of reasonable safety for anybody, any normal human being's definition of reasonable safety, research grade lipid nanoparticle mRNA seems pretty reasonable. I mean, I'm not just saying that as somebody who's running a company and going to stick this into other people. I have had a research grade gene therapy of that nature, and that seemed like a very reasonable risk to undertake, given what I know of the way these things are manufactured.
Yeah, so $20 million. So one of the things we're going to be doing with this roadmap is we're not building a technology tree the way Foresight was. What we want to do is have a list of specific projects with time and cost estimates. And I think yours would be one of the ones that we would want to include. And we want to help you get that money, basically, either by raising it ourselves or just inspiring somebody else to fund it. But I'm kind of curious as to, obviously, you've probably pitched a lot of VCs at this point. Do any of them ever say why they don't fund you? I mean, I know VCs tend to not like to actually say why, but like, what do you
You
think is
always
going
get
on?
the weird answers when you ask these questions. Though occasionally you luck out and an associate leaves, and you can chase down the associate afterwards and say, "Hey, okay, now you're not working there. Give us the real story." And actually, one of those was the very fascinating reason that like the math just didn't work out. They couldn't put the right amount in to make their model work, and it was nothing to do with us or anything else. It was just weird model silliness. But VCs are like this. I mean, this happens. So, I mean, there's several stages at which we've talked to VCs in the course of this whole exercise. There was a period in which we were pitching, attempting to treat homozygous familial hypercholesterolemia with AAV. So use AAV to deliver our gene therapy. And on the face of it, this is not an unreasonable proposal, because other people have tried to use AAV to treat homozygous familial hypercholesterolemia by correcting the gene, the LDLR gene that's lost. Unfortunately, that doesn't work for reasons that are too complicated to go into here, which is very sad, because that closes the door on the most logical, sensible approach for these patients. But at the time we were doing that, the VC community, this was immediately post some deaths that had occurred in trials of high dose AAV. And at the moment, the FDA and investors are basically going to tell you no if your idea is high dose systemic AAV for any indication, although they're still friendly to low dose localized AAV, and that's still sensible. But the current thinking is that nobody's really interested in high dose systemic AAV, and that's what would be needed in the case of what we're doing. So we tried that at the time, didn't go so well. And that's one of the cases where we actually fully understand why it didn't go so well. It's a risk aversion point of view. The other things that have occurred over the years is that if you're doing a first-in-class therapy, if you're targeting a new mechanism, particularly if you're targeting a new mechanism in an industry that's very ossified around an existing mechanism. So, and this goes back to surrogate endpoints are the devil's bargain. They really are. As soon as you have a surrogate end, as I like to say, as soon as you have a surrogate endpoint, all original thought ceases. At which point the disease is now that surrogate endpoint. That surrogate endpoint is the be all and end all of what you can do. Every investor only understands the surrogate endpoint. If you come in and present them something new, they do not know how to evaluate it. And that's an automatic no right there. Unless somebody else leads, in which case they're like, sure, thumbs up, let's go for it. The trick is always finding that savvy investor who's prepared to put in the assessment and trusts their scientific advisory staff to the point at which they can evaluate a first-in-class therapy and know what they're talking about and know what they're doing. And that there aren't many of those. And I will tell you, a lot of the very big investors just are not capable of evaluating first-in-class therapies, or don't feel themselves to be capable of evaluating first-in-class therapies. So they will waste your time, but they will never invest in you. And that's been a problem, trying to convince people that you can tackle atherosclerosis without doing anything about blood cholesterol. That atherosclerosis, in fact, does not depend on blood cholesterol. The blood cholesterol is not the important mechanism, that the whole research community has been barking up the wrong tree for the last 40 years. And that's why they only have 20% reductions in mortality. That's a hard sell, unfortunately, just because of the way the community is set up. And there's no shortage of people out there making new drugs to lower LDL cholesterol in some different way for some different group of patients, or do it faster or do it different. And those guys get funding, even though they're not really actually changing anything.
Yeah, I actually can think of some investors that I think might be amenable, but maybe we'll follow up on that afterwards.
Well, you absolutely should. We just got our first pre-IND package, like sort of still have to dot some I's, but it's basically done by the end of this year. And then we're immediately going through our whole Rolodex and working on an A round.
Let's follow up on that. And I think maybe now is the time to switch into, I think, Nathan's favorite topic, which might be interesting conversation to have here, since you mentioned an aversion to surgery.
Oh, yes. So Reason, what do you think about whole body replacement?
Yes, whole body replacement. Well, that's saying I don't want surgery so hard you wrap around all the way back to surgery again, right? So, I mean, I think the whole body replacement thing is technically interesting. I don't see how we will ever in this environment get significant funding for this. Though, I mean, cryonics kind of got over a lot of the stigma, but that took a long time. And I think like the as soon as you get, I can envisage the press articles already. As soon as somebody starts earnestly investigating how to grow bodies without a brain, for the purposes of organ harvesting or transplantation.
We like to call them non-sentient humans.
Yeah, I'm not sure that helps. This is because we have so much literature on this recently. There's at least two major properties in the last 10 years. There was the film The Island, and there was around the same time, there was a piece of literature from a fairly famous writer that touched on the same thing of harvesting humans for organs. And people see this. People have. I mean, you know what people are like. They if they threw a fit over embryonic stem cells, they're going to throw a very big fit over, or and genetically engineered children. They're going to throw an even bigger fit over let's make bodies. Even though at the end of the day, if the capacity existed and people were using it, everybody would say, "Meh, it's just another part of medicine." But actually, this would be a high risk project to put money into, I feel, because of the likelihood for blowback at this point in time. But okay, let's put that aside. Scientifically, it's interesting, it's plausible. The objection comes when you start to say, okay, so how much work would it really actually take to get a brain transplant going? So head transplant, that's less. And even there, I feel that like that is tough. There's actually a lot of work to be done to make that something that would be viable, reliable, that would work well. And even if you did a head transplant, you're still looking at a very aged brain. And we still need to figure out how to fix everything in the brain, because transplanting to a new body is not going to do that, and conceivably could actually cause damage because of the change in circulatory patterns and strength and all the rest of it. So brain transplants are still, I would say, 20. If we got like a real industry that was working on brain transplants, just looking at the speed of the xenotransplantation industry, I'd say we'd be looking at 20 to 30 years before anybody tried anything in humans. And then it would be another 20 years before it was a going concern. So I think it's hard. I think that's harder than most of the people who are involved in this think it is, or estimate it to be. I think they're very seduced by this idea that you can bypass all of the hard work you need to do for in situ rejuvenation by getting a new body. Except from my point of view, a great deal of that work is actually rejuvenating the brain, which is going to be a lot harder than the body. So I feel that it doesn't gain enough, and it presents challenges in and of itself around the surgery part of it that are equally as large and likely to be equally slow as are facing rejuvenation focused on the body itself. If that's that, I think that's a fairly high level comprehensive answer.
Yeah, I guess I'll challenge a little bit on that. Like, do you really think the complexity of aging versus the complexity of refining a surgical process for head transplantation are on the same order? Like, my intuition is that the biggest obstacle to replacement is actually social stigma, and that from a technical standpoint, getting the transplantation process to work is much easier than solving all of the, I mean, forget the brain temporarily. We can get back to the brain, but just the technical challenges of getting body transplantation to work. Intuitively, to me, that seems much simpler than solving, coming up with a cocktail of therapies for all of the types of damage that accumulate.
Okay, I'd buy that. I was wrapping in the brain part of it a little too much. The brain part of it really says to me that, you're kind of stuck because you need to fix the brain. fixing the brain has that look to it, like fixing the immune system, that the only way to do it is going to be fix all of the underlying causes of aging. And if you don't do that, then you're still left with meaningful neurodegeneration. That's my feeling looking at the literature in any case.
How deep have you looked into Jean Hebert's work, I guess?
I know what he's doing. The whole replacing parts of the brain are amenable to replacement. Anything that's not actually holding your state of mind. If we assume that state of mind is stuck in certain parts of the brain, in the dendrites, or inside the cells themselves, if we were to believe the sea slug research, then there are entire pieces of the brain you could just hack out and replace with other pieces of the brain. But even there, that sounds like a tough job because surgery is tough. And for preference, I would not want to go into a future in which, you have to undergo 6 major surgeries every other year to replace pieces of yourself, because major surgeries have at this point in time and for the foreseeable future quite a high mortality rate. It's like a few percent for many of them. And if you're old, that goes up. So it just doesn't seem good compared to spending the effort to actually do in situ repair in some way.
Maybe another way to think about this would be if you were placing bets on different strategies. So there's a lot of people that are proponents for replacement. The largest segment is small molecules right now, which I think you think is vastly over invested in.
Mm-hmm.
Currently, replacement is, I would say, getting very little investment. I mean, if you were to rebalance where we were distributing funds and resources, do you think that replacement should get more relative than it gets now, like as part of that rebalancing?
Well, I mean, if we want to take replacement as a much broader view that includes delivery of cell populations, I think that one of the most important problems that could be solved is how do you replace a stem cell population? How do you deliver new stem cells to replace one of the stem cell populations? I mean, we have ridiculous numbers of different ones, but that's a problem that needs to be solved and hasn't been really cracked. The survival rate of transplanted cells is terrible. Getting them to engraft is really hard. And outside of hematopoietic cells, nobody seems to have made much progress on this yet. There are many cell populations that are sort of near the top of the list, like muscle stem cells and neural stem cells, that you could probably get a great deal of benefit out of replacing if we could just figure out how to do that. More funding for that would be a good plan, I think. Definitely other forms of replacement, everything starts to look like surgery. And I'm less in favor of that than for making gene therapies better to the point at which we could argue that we could deploy reprogramming therapies more effectively in vivo and or other approaches in vivo that would clear senescent cells and fix some of the lipofuscin issues and reprogram specific cell populations to go out there and behave more like they were when during development, like cells responsible for elastin. We can't have the future of extracellular matrix therapy be here's your highly targeted EDTA chelation therapy, or go take ultra high dose injected minoxidil to make some of your cells behave more like they were when they were laying down elastin during your development process. It has to be something better than that.
Yeah, so you're brought up the extracellular matrix, and I think that's been getting a lot more attention recently. You also mentioned the fact that cell therapies often have a pretty poor rate of uptake, that they just get cleared. And so I think there's some people that would say that the aging of the extracellular matrix is actually the problem, because you can put a young cell into an old body, but because it's an old body and there are these long-lived structures that are aged, the changes to the cells are actually that make them seem older are actually adaptations to the aged extracellular matrix. And so like they just quickly just become old cells, even if you injected them as if they were young cells.
Well, there's a lot we don't understand, and that could be true or not true for different cell populations to different degrees, right? And it could be different problems in the extracellular matrix, and it could be nothing to do with the extracellular matrix, and it could be to do with the signaling environment, and it could be to do with the fact that the cells just are never going to play ball in their current state and you need some kind of different cell or reprogram into the one you want when you get them there, I suspect it's going to be different for every stem cell population as to the degree the extracellular matrix is involved and what the problems are. But certainly the extracellular matrix itself is a thorny issue. What do you do with it? And I suspect that the only viable answer in the near term is going to be find ways to reprogram cells to better maintain it. Because everything else looks kind of fiddly if you're saying, well, let's build a company to target a single form of stuff that doesn't look right in the extracellular matrix, like elastrin, who's just getting rid of broken elastin. You can't build 100 companies to go do this. You need something better, and that something better, I suspect, is reprogrammed cells.
The Kerberg initiative has just started what they call the engineered cell program, and I think one of their targets is to make modified macrophages that could go in and do things like that, basically. This is your nanotechnology, not building synthetic nanotechnology. It's building engineered macrophages.
Yeah, macrophages are microglia analogously, because microglia are basically macrophages
for the brain,
the central nervous system. There's definitely this very interesting. They look like a low-hanging fruit in the sense that they run off the rails and they go all inflammatory. And if you could just stop them doing that, then maybe they would just get back to repairing tissue and doing things and making things much better. And there's plenty of evidence in liver-targeted therapies that this is true. And plenty of evidence for the brain that this is probably true too, though it is inordinately complicated. That's also the challenge. I mean, if you're putting macrophages in there, you're kind of trusting that they're going to go off and do sane things. And if you get it wrong, you get fibrosis, which can't be reversed at present. So that's kind of, it's a challenge, but it's also a very promising area of study. Agreed. Macrophage therapies. So we actually looked at that. It turns out that our approach to making cells eat cholesterol makes macrophages happy. We did actually look at this at a point in time, but it's just not cost effective. At this point in the development of the field, working on cell therapy is extraordinarily difficult and expensive. And major companies like Sana have stumbled after spending enormous amounts of money on trying to get somewhere. It's very hard. And the successes are sort of, when you look at the successes in cell therapy, it's maybe easy to say, well, we should be doing more than this. But the few successes exist because they just happen to be somewhat easier than the other options. And almost everything else people have tried. There's a reason why we're still doing trials in Parkinson's patients for replacement of neurons. And it isn't, a widely available therapy. This stuff is hard. I mean, you can't possibly. How hard can it be? We're putting neurons in a brain. And actually, it turns out that even that very simple proposition is very hard. So tough business, cell therapy. And I don't expect it to stop being tough anytime soon, which is another reason why reprogramming of existing cells is a more favorable approach than anything involving trying to deliver cells.
Although ultimately, I think engineering cells outside of the body. So if you want to give cells a fairly complex new behavior, doing that engineering outside of the body and delivering it has advantages. And that's where engineered macrophages or engineered microglia could be actually very promising.
And the reason why it does is so anybody who's engineering ex vivo, if they could engineer in vivo, they would do that in a heartbeat, because it's much cheaper, much easier. The only reason they're doing it ex vivo is because you can't do it in vivo for their particular circumstance. This comes back to gene therapy vectors being not a solved problem, and there's very good chance that there is no vector to deliver exclusively to your cell population of interest.
So we already covered replacement and whole body replacement, progressive brain replacement as well. So that's one of the approaches, high level approaches that we've put on our roadmap that we're interested in diving deep on. And I guess we're kind of also discussing gene therapy, cell therapy, which is in the third category, this bioengineering strategy. But we can get deeper on that later too, return on there. But I do want to give us some time to talk about so cryopreservation. What do you think of the state of that right now? And should more people be working on this? I know in your blog, you mentioned sort of a deadline, a condition in which you would actually spend more time working on cryopreservation.
Yeah, we're getting there. Time waits for no one. So I think it's necessary. on the grounds that it's better to be alive than dead. If alive means my brain state is continued to be stored, and there's some chance that that brain will be thawed and resurrected in the future. But equally, you got to make your choice here as to, is it better to keep hacking away at rejuvenation that will benefit more people? Because cryopreservation is a long way from any sort of widespread adoption. In a sane world, yes, everybody would be getting cryopreserved. We obviously don't live in that world. And in a sane world, a lot more resources would be devoted towards how do you properly cryopreserve people and reverse that cryopreservation. And I think the small industry has made very large strides in the last 10 years in terms of new people coming in, having new ideas, trying to bring fresh blood to the space and speed things up a bit. The most important development that is still somewhere in the state of getting to happening is developing reversible vitrification and applying it to the organ transplant industry. Because if you can, that seems very feasible. And if you can do that, then you can convince a lot of people that if it works for an organ, it'll work for a brain and a whole person. Maybe we shouldn't be letting people rot in the graves or get cremated when they should be getting preserved in large stacks that are going to be stashed somewhere in low value land or put into orbit or wherever they end up being, because it's better to be alive even in that state than to be dead. But we don't live in the rational world. We live in the world in which incentives rule. And so the best possible near future approach is to take techniques for cryopreservation and thawing that are in very nascent stages and turn them into a company. Maybe that's what I'll do next if nobody's done it already. Though on the time scale I'm looking at, I would say that it's quite likely somebody will be doing it already by the time I get there, which is good.
What have you thought about what you would do? What part of cryonics would you work on?
I think like the part that I would be interested in doing, if I wasn't doing reversible vitrification, I think that the Europeans who are currently working on making it more friendly to the public, that's a good place to be. It's a good
You're
place
talking
to be
about Emil?
Yeah.
Consumer.
And their efforts to make this more of a viable consumer business, where you're actually, stopping it from being, take away the stigma, make it a sensible life choice, like 401ks, that sort of thing. That is somebody needs to do that, because there's bootstrapping going on, right? Well, I'm a technological determinist in the sense that if you have a capability, if you can make a capability, then people will come eventually, even if you have to shove it down their throat and explain it to them 20 times, eventually they will come. There still needs to be that other side of the house where you're advancing the way in which it's marketed to the world. So it as I said earlier, when you're talking about, okay, if I'm coming into the cryopreservation space to do a thing, what things are there technically that needs to be done, and are there research programs out there at a ready stage? Because otherwise, you really have to either have philanthropic funding or raise philanthropic funding to run research programs to get it to the stage at which it can actually start launching a community and an industry and so on, right? The reversible vitrification is the best way to go about that, but it's unclear as to when that's going to launch. We'll see.
Do you have any thoughts about cryobiology? I just have the intuition that nature has actually solved this problem in the sense that there are organisms that can be frozen and thawed.
Yeah, and we're not one of them, not easily anyway.
Yeah,
so,
but then it makes me think that the gene delivery problem may also be the problem that cryo has.
So that's actually a subset of the interesting field of the comparative biology studies. There's people who are looking at salamanders from the point of view of, can we make humans regrow legs? And people who are looking at elephants and various other low cancer entities to say, can we make humans stop having cancer? And I'll say, I think it's probably going to be extraordinarily difficult to re-engineer the human genome, even in simple ways, to introduce new capabilities. because this is stupidly complicated. I don't think anybody is capable of predicting what a new gene into humans will do in all of its ways. You can't just change something and expect to get somewhere. This is one of the again, back to one of the foundations of SENS, is that trying to change human biochemistry is stupidly complicated, whether you're doing it with a small molecule or you're introducing some different gene. Whereas editing it messing with the body to restore a known good state, such as by removing senescent cells, is something where you can expect some confidence that you're going to do good and that it's not going to have weird effects. So if you're talking about, there was recently a study in which a group of researchers took the hyaluron gene, one of the hyaluron genes from naked mole rats, and put it into a mouse, and the mouse lived a little bit longer. And I honestly don't think it's going to be that simple for any of the good stuff to actually bring it into humans, and certainly not if we're talking about taking genes from frogs. There's a lot of problems that have to be solved to do that. And one of those problems, the biggest first one, is that nobody will fund you to introduce novel genes into humans. They will say that will never get approved by the FDA. And they're probably right. The problem is that the FDA's view of immunogenicity in therapies pretty much excludes any introduction of a novel gene, and even for like slight modifications of existing proteins, you have to go through very stringent tests. So somebody certainly could, in principle, produce a novel gene, put it into do all those stringent tests to suggest that it's not going to cause immunogenicity. At the end of the day, that is going to be a flat no from almost every institutional biotech investor that you talk to. They're just not going to fund you for that.
I think in solving the immunogenicity problem is going to be necessary, though, ultimately, no matter what, like, right? Because we're talking about the idea of eliminating all aging altogether, and just the sheer quantity of, like, even if we're doing the gene therapy approach and the damage repair approach, you're still introducing probably new enzymes, things like that, right? Like, and so this is where, like, I think the engineering approach and the replacement approach can work together, right? Because we do have solutions to tolerance, right? Bone marrow transplantation is a thing that actually works and allows you to establish tolerance.
Bad solutions to tolerance.
They're not. Okay. Yeah. Here we go. Tell us what's wrong with tolerance.
In the sense that, do you really want to undergo a bone marrow transplantation every time you have to do something meaningful in your biology, or get your thymus injected, which is another path to tolerance?
If it's every 10 years, then it keeps you alive.
But at some point it stops being cost effective. In any case, my point being, there's a lot of challenges involved in the idea of introducing novel genes and novel capacities mined from other species. It's not to say that there may not be interesting options. If you find that a particular gene is problematic, you can always suppress it, for example, in a certain circumstance. But it's not clear to me at all that any of the comparative biology of aging and regeneration will, in any near future sense, deliver things that are meaningfully useful to humans. We'll see. Maybe I'll be surprised. But yeah, I mean, and on the cancer front, you can't just do the elephant thing and put in extra copies of P53, because
they did that in some mice, didn't they?
Yeah, and to a certain it's if you upregulate these anti-cancer genes, you get shorter lifespan, more tissue failure, because there's this balance between cell activity.
Yeah, you can't. If you just put more copies, you end up with more of the protein expressed. But what you actually really want is extra copies, but with the same level of total expression, which is more complex.
Yes, and that's the motto here. More complex. That's always what's going on here. It's always what's going on in the life sciences. It's always more complex than you'd like, which is why I suspect that the comparative biology may even though it's delivering some very interesting information, I think it's going to continue to struggle to deliver actionable items that you can then use for therapy, even in what looks like the simplest of propositions, which is, can you undo the restrictions on adult regeneration in a mammal? Because we have all these examples of mammals where that restriction is relaxed for some tissue, or it's relaxed due to some gene manipulation, and examples where particular genes from mammals appear to suppress this activity in killifish or zebrafish or other entities. And we have examples of information that the interaction between senescent cells and macrophages seems to be really important in this proficient regeneration process that's going on in salamanders and zebrafish. So there's all these pieces of the puzzle swirling around that suggest, and we know that young children are capable of proficient regeneration, and embryos are capable of it. So surely one might think there's a way to just switch off whatever suppression is going on in the adult and do it in a way that doesn't give you infinite cancer. And maybe this is that simple, but the cynic says, I doubt it.
Yeah, the strong proponents of cellular reprogramming, I think, are the ones that would say that. But then, yeah, I don't know if we want to get into that one on this episode. That's a deep discussion.
Reprogramming is interesting. We can talk about it.
Do you want to tell us what the challenge is for reprogramming? Because we are going to be bringing on some of the strong proponents of reprogramming on the show. So I'd like to hear what you think of it, I guess, then.
So I think I'm very optimistic, personally. I think it's great that it's turned out this way. And the important thing that I'm waiting for is validation of this work that double-strand DNA break repair causes epigenetic change. Because if that's validated and true, then that means that epigenetic change is actually a lot closer to the root of aging than suspected, which means that reprogramming is actually a great therapy. And it's not trying to push the boulder uphill. It's much better than I thought it was going to be. We'll see. But the challenge for reprogramming is the gene therapy challenge writ large, which is, how the hell do you deliver something that has to be different to every single cell type it goes to? And that's tough.
And maybe to every cell, not just every cell type, actually, is what I worry about.
It's going to be tough, I think. I think there's going to be some wins that are easily done. For example, there's some people turned up an AAV variant that has 30x tropism to skeletal muscle tissue. So that sort of thing is great. And maybe there's enough of that one could do early reprogramming to tissues that are accessible, and get some benefit out of that, and start stone souping your way to more therapies from there. But it is delivery. Delivery is entirely and completely the challenge here. In that, because mRNA basically works. It gives you the right sort of level of expression. It's a good technology for this. The trick is getting the mRNA to where it needs to be. And that's going to keep people occupied for a while. And I think there will be better technologies coming on after that, such as plasmids that can be disabled at the time of your choice, which are more amenable to controlling expression in different places according to promoters and other such restrictions. But reprogramming really honestly looks pretty good. It gets good results in mice, and certainly one would hope that those results also show up in humans. And why wouldn't you want more youthful cells if the risk of cancer doesn't appear to be appreciable?
Cancer reminds me of another topic that we'll be getting into some great depth with some people, which is nuclear DNA mutations. I think there's been an ongoing debate as to their importance for aging, like the somatic mosaicism, for instance. Cancer is one of the results of nuclear DNA mutations, although you can even argue that the nuclear DNA mutations themselves are not necessarily the biggest driver of cancer. But yeah, how do you think about, because I remember way back when I first started reading your blog, that was one of the big debates and the few forums that existed. It's like, how important are nuclear DNA mutations? Because if they are important, that certainly presents a challenge.
I think the somatic mosaicism crew have managed to improve on the state of their data over the last 10 years. I mean, it still needs further improvement, but I think it's fairly safe to say that somatic mosaicism is not great. And it's a contribution, though as for everything, nobody can measure exactly how bad it is versus other things. But I think somatic mosaicism is something that we want to get rid of. And of course, the way you do that is by replacing your stem cell populations, which is why that's an important focus. And then you wait and your somatic mosaicism goes away over time. The place that's really important for DNA damage, I think, is the brain, because for the same reasons as everything else, you can't just replace much of it because it's storing you. So that's a tough problem, and I don't know what's going to happen in the future with respect to that. How do we deal with that situation where you want to keep cells, but you have to repair stochastic damage to those cells, or you have to in some way compensate for stochastic damage to those cells? This is why the brain is sort of the final frontier of all of these things. And none of the strategies that we adopt based on some form of replacement can be applied completely to the brain. So people 50 years from now will be thinking hard about this, I would imagine, but we have so many other problems before then that we can just proceed and hope that the answers fall out of what we're doing along the way. But I think it's much less clear than it was 10 years ago that one can just say, man, nuclear DNA damage throughout the body, cancer, and it's only relevant to cancer and nothing else. I think there's decent evidence now that somatic mosaicism is in fact a problem.
Yeah, and I think in the brain as well, for instance, like retrotransposon activity goes up exponentially with age, which is potentially a result of the epigenetic aging.
Yes. And that of course means that the earlier you solve the epigenetic aging, the less DNA damage you have to contend with. Though of course near all DNA damage occurs in cells that don't matter, or in areas of the cells that don't matter. It's largely the fact that some of it is occurring in cells that replicate over time is the problem.
Yeah. Okay, great. So there's one last part of bioengineering that we wanted to ask you about. Part of the problem on bioengineering, like gene delivery and trying to modulate human biology in a significant way. But then the other half of that particular strategy is trying to figure out just what to modulate, what model do you have to understand aging or biology writ large. The wild card here is maybe artificial intelligence. We've been talking a lot about humans have to understand all this complexity, and it's just way too complex. But there's obviously some developments going on, very, very salient to our conversation now. I'm curious, what do you think about AI? Does it give you hope, or does it inspire terror in you?
Well, it's massively overhyped right now. So obviously it's in its early stage of the hype cycle, and people who understand hype are doing their job very well. So I think that the sane place to look for what is presently called AI and what should actually be called automation is in drug discovery. Because if you look at what has to happen in drug discovery, which is search for mechanisms, search for small molecules, then this is a lot of very detailed work with enormous data sets, which is what software specializes in. So machine learning techniques are another misnomer, because the machine is in no way doing something that could be called learning. These things are best applied to that sort of problem, and people are applying them to that sort of problem, and it cuts the cost dramatically. They're an infrastructure technology that allows you to do more with less. Now, to the extent that it's being used by people to pump out a bunch of garbage drugs where they're just saying, oh, look, a mechanism, let's see if we can tinker it a little bit. Oh, look, we got a drug that slows aging a little bit in an animal model, go for it. To the extent that these are being used to pump out yet more garbage drugs, then yeah, that's not going to do an awful lot for anybody. But equally, the same technology is going to be applied to senolytics, which are not garbage small molecules and do actually have useful effects. That's where these things are useful, in taking the parts of drug development that are actually very data intensive and trying to process it faster than people can, to get answers out of it that are sane faster than people can. Other than that, honestly, At the company I run, zero automation. Not needed. We have a mechanism. We know it works. Everything is wet lab. We barely care about computers. Computers are things that you stick on top of a piece of lab infrastructure and make it run. This is not the way that a lot of these groups work, because at this point in time, if you look at where the money is coming into the field, a large amount of that money is coming in from the traditional investors who know tech. If I had a dime for every fund that only invests in the intersection of software and biology, they're doing this because they know software. They're not doing it because it's useful. In fact, it isn't particularly useful in the grand scheme of things, I believe. It certainly helps make drug development faster, but most of that drug development is garbage. That's kind of my take on what AI is doing in this field. There are certainly areas in which it helps, but AI doesn't help you pick the right problems. It doesn't help you choose to work on the list of SEN strategies. If you've chosen to do something mitochondrial rejuvenation, and then you find that there's an enormous sea of data that you need to make sense of, great, go use AI. But, if you're just doing unbiased searches for ways to modestly slow aging and just have another Me Too drug that follows on the coattails of rapamycin efforts and bioage, then, the world, whether you exist or not, makes no difference to the future of the world.
I think you're correct in terms of like the current state of artificial intelligence, which Joe Lacroix would just call software. He wouldn't even call it artificial intelligence. He would say that's a misnomer. It's just a way to crunch data. But there have been like sort of step changes, and I think we have seen like a step change recently in capability with AI. And I mean, you're imagining in your model of how we escape aging, or how you personally escape aging, is most likely cryopreservation, and then a bunch of work is going to be done, and then we'll be able to wake you up at some point. And we've also talked a lot about like the sheer complexity of aging. So do you see like future iterations of AI being part of that? What makes it possible to actually wake you up later? Because I kind of suspect myself that if we're doing human-based wet lab work, we might hit some limits on what we can achieve with human level intelligence.
Well, at the time when we have a fully automated lab which is run by an AI that can request reagents, supplies, I don't think it's going to make much difference, because garbage in, garbage out, and what we have in the literature is substantially garbage. I mean, we know this. So in the future, my feeling is that aging is not going to be solved by discovery. It's going to be solved by taking what we know are the causative mechanisms of aging and exploring around how we influence them in order to produce effect. And humans can do that. Machines are only going to help on the low-level details of what of the 10,000 ways to produce an effect here should I really be spending time focusing on. But even so, they can't do discovery. That's not discovery. That's mining the existing literature and mining the existing databases. If there's something new, AI can't help you with that, and probably will never be able to above and beyond human capabilities, because you still have to get out there and test. So at the point at which you're producing automated labs that form and test hypotheses, then I think that AI is relegated to these areas where you suddenly run into a dense forest of data and you need a quick way through.
Yeah, I think that's the current state of it. But the market incentives to produce ever more powerful AI are very strong. Like that's one of the biggest problems we have in aging right now. We don't have the market, we don't have the stories, we don't have the right focus, but there is like massive resources being driven towards advancing AI right now.
I would dispute that. I think AI in the sense of general AI, no, there are not actually massive resources being devoted to that. What there are massive resources being devoted to right now is optimizing the spam industry. So everything, all of the funding around what is currently called AI exists and is directed to enabling people to produce, quote, content, unquote. Because we have this massive snake devouring its tail industry of attention and garbage being produced on the internet. And that's all this is. That's all this is. The only possible applications for AI in its present sense are producing garbage faster, outcompete other people producing garbage. The people who are actually interested in doing useful things with this, tiny minority. And they're not the ones driving the conversation, and they're not the ones with the money paying for the services. So,
well, even when they are interested, their incentives are to compete with each other for short-term business wins.
yes.
Right. Like I think like Dennis Hassabis, like they from DeepMind, and even Sam Altman, like have expressed an interest in the applications for science. And Google in its early days was trying to support scientific research by making free databases and things like that. And they dropped them just because they are corporations driven by quarterly profits, and
there
they
is
realized
the application to research, honestly, outside of what's already being done, which is to manage the enormous outgrowth of omics data. What can an AI do that a good human can't do in terms of ideation to go do the next thing? It can't. The things that are not written down, the things that are not in databases, that is what is important here. And AI can only regurgitate what already exists in a form that makes it easier for a human to understand, which is not unuseful, because we do have ridiculously huge omics databases that are just impenetrable forests of data and need automation to make some head or tail of. But again, this is not the AI that's going into turning the internet into a wasteland of low effort content. And that's where the money is, and that's where this will all go. I really do not believe that we're going to see any sort of progress to general AI emerge out of this cycle. All we've learned is a cute trick about how neural networks can work in this instance. And at the end of the day, I strongly believe still that the path to general AI is reverse engineering the human brain. You scan the human brain, you figure out how to emulate it, and that's going to be the first forms of artificial intelligence. And after that, a whole load of exceptionally unethical stuff is going to happen, just the same as it would happen, frankly, if AI was generated some other way that wasn't through humans. And I'm rather glad that I will probably be asleep for most of that.
what gives you hope?
The distant future, I think. In the short term, people are idiots. Most people don't care about improving the human condition. And yet, in the longer term, the trend is clearly up. Everything's getting better. Everything continues to get better. We continue to have new choices. Things continue to be less horribly terrible. So I think if you look at the distant future, our descendants will be a starfaring explosion of sentient entities that are immortal, moving as close to the speed of light as they possibly can, and obtaining as much of the light cone and turning as much of that as possible into sentient matter. And of course, a seed should not be afraid of turning into a tree, as the saying goes.
I like the light cone reference because, Elon will wear a shirt that says Occupy Mars, but I think that's not ambitious enough. We need a shirt that says Occupy the future light cone,
Mm-hmm. Yeah.
Yeah, but when you say distant future, do you have any estimates on how far that is?
Oh, God, no. I mean, if you want to look like an idiot, predict the future. So I have no idea how all of this is going to happen. I only know that regulated medical development is very, very slow. And also that the path to getting into orbit appears also very, very slow. But at some point, we will have low-cost orbital transfer, at which point the whole solar system becomes open to us. And after that, inevitably things will grow. So the future will happen on some time scale. And along the way, all the important capabilities that lead to the vision I just outlined in brief will happen. Our descendants will be immortal of some sort, likely not very biological, and they will conquer the universe.
So another maybe speculative question. Do you think there is a path that doesn't break the law of physics, some path in the multiverse or whatever you want to call it, where aging can be solved given the resources and the constraints that we have now? Or do you think within, I should specify a timeline, maybe within 40 or 50 years?
Well, within 40 to 50 subjective years, the answer is cryonics. Let's just go away for a while and let people sort it out. Other than that, no, clearly this is too hard. It may be that there are surprises. It may be that one of the things that people are working on in the SENS list turns out to be actually really important, and there's one little linchpin that gives you 20 extra years. I'm not counting chickens on that, and I don't believe it. But equally, everybody's being consistently surprised by what happened in mice as soon as you start doing things. So who knows? Maybe that will happen.
So you're not as optimistic on this sort of like nice, smooth LEV rollout?
No, I think it will be a very uneven rollout for the reasons given. Regulation makes things very slow, even though it does tend to bend when amazing new capabilities come along. And also, we have absolutely no idea as to how good all of these things are relative to one another. So, because they're all happening at once, and typically you can't address one in isolation. The only one we've really done is senescent cells. Everything else is still waiting to be challenged, and senescent cells turned out to be a lot better than thought. But equally, after all of that, if it extends life by 35%, 36% in mice, what does that mean in humans? Nobody knows, because there's no point of reference here.
Does it concern you, like the 30% number, that was like the median lifespan extension and like the, those seminal 2016 papers at
Yeah,
the
that
Mayo.
one.
They didn't show any maximum lifespan extension. Do you make a distinction between that?
It's very clear that mice do not age the same way people do. And that's not just the different duration of life. It's that there's very different collections of processes and where frailty fits in and frailty doesn't happen. Humans just age completely different. If two species age differently, they age differently. And it's very hard to draw ideas about, okay, so, if you put mice to jogging, their median lifespan extends. If you put humans to jogging, we know that life expectancy goes up by 3 to 5 years. But what does that mean for total life expectancy? I don't know. Nobody knows. Nobody's done that study. If you calorie restrict mice, 40% extra lifespan. Calorie restrict humans, we know that doesn't happen because it would have been known for thousands of years. But equally, we have no real idea what happens to human lifespan because nobody's done that study. Growth hormone knockout extends life in mice by 60 to 70%. In humans, definitely not, or we would have known that because there's a human population who have that loss of function. The Laron syndrome dwarfs do not appear to live longer than us. They may be more resistant to certain age-related diseases, but again, that's still up in the air. But the effect sizes here can't be large. So there's this thought that, and then, okay, these things, growth hormone and growth and calorie nutrient sensing type stuff going on, this is completely different from senescent cells, which is more of an inflammation, tissue dysfunction problem. So maybe it's completely different. Maybe in humans, you get 10 extra years. We just don't know. We've been into this whole senescent cell thing for 10 years. And it's still the case that it's going to be another 10, 20 years before anybody even starts to think about, let's churn some data because we have enough people taking this drug to start to say, what's the effect on life expectancy in humans? Who knows?
Yeah. And that's the thing, like actually demonstrating lifespan extension, you have to measure it. Although some people want to use, aging biomarkers and stuff like that. Do you have any thoughts on that?
They will work at some point. Any sufficiently complex set of data, it's going to reflect some amount of aging, underlying aging processes. If you have an epigenetic clock that is based on 4 CPG sites or 7 CPG sites that does a really good job of correlating with chronological age and appears to predict biological age, I think we can reasonably say that that thing will fail horribly as soon as you start throwing interventions at it, because 7 CPG sites can't possibly represent a digest of all the relevant underlying processes of aging. So maybe it isn't even measuring and we know that the first epigenetic clocks were insensitive to fitness status. So obviously that's just wrong because we know fitness status slows aging. So, those clocks need a lot of work. One of the things the community needs to standardize on a clock, which will probably happen at some point, and then test that clock with everything. So any therapy you do, you test it with the therapy in question in parallel, and then you can use that clock to assess other therapies of that type. But until you've done that work, you can't trust any of that data. So there is no shortcut. In other words, people should be testing epigenetic clocks with everything they do. but they shouldn't be running off and testing their own or making new ones. They should be testing GrimAge and the standardized clocks. It can only help to have more data that says, oh, look,
Yeah.
wait, if I do this with my therapy that slows aging in mice, nothing happens in the epigenetic clocks, or something happens, because then at least you get some data on whether these clocks can be trusted.
We were just at the Biomarkers of Aging Symposium at the Buck, and Kristen Fortney brought up an interesting topic, which was when they're doing their research, they try to measure everything possible. But when people do a clinical trial, they try to measure as few things as possible because they don't want to demonstrate a side effect.
Yep.
But what we would actually, if we want a clock that works in the context of interventions, this is a case where AI can be useful, right? If we collect masses of amounts of multi-omic data on people aging, but also people in the context of every possible intervention that you can think of, and maybe even do perturbation studies where you, right? Like that is the kind of thing that could eventually give you a biological age measurement that you could believe that might actually mean something. And that's a massive project. That's like the Human Genome Project, right? Like that's what we have now is a bunch of, I would say, piecemeal, small projects where people are studying certain types of clocks. But what we really need is something like a Human Genome Project level. Let's look at everything in as many types of people with as many genetic backgrounds, look at biobank samples from every clinical trial we can get our hands on to see how those drugs affected aging. To me, that's what we would need to do to actually get a biological aging clock that was validated and meaningful.
Yep. And people like Jero are sort of baby steps in that direction, the analysis that they are doing. But it's a big challenge. And I think that the only thing you can do in the meanwhile is just show that actually, yes, you're having effects on specific age-related diseases.
Yeah, I really like what Jero is doing.
Yes, I think that's good. You know, I'll definitely thank you a lot, Reason, for having you on as one of our early guests. You've been a figure in this industry for a long time. Anybody who has not seen the Fight Aging blog should go check it out. Any investor who's listening to this and is not afraid of getting into a new modality or a new way of thinking about disease and wants to potentially have a piece of something that could be game-changing, should definitely talk to Reason.
Well, much appreciated. Thank you.
Is there anything else the audience can do to get in contact or help out with your request?
Always just reach out if you have ideas, Fight Aging or Repair Biotechnologies. I'm amenable to being contacted.
Okay, great. Awesome. Thanks, Reason.
Okay, thanks all.
Bye.
Thanks a lot. Have a good night.